The lab you are doing today is the ELISA test. Before you start the lab, it is important that you must pay attention to the order of adding solutions. You also must use a new micropipette tip after each use to prevent cross-contamination. ELISA stands for Enzyme-Linked Immunosorbent Assay. During this test, you'll be testing for HIV indirectly by testing for the presence of HIV antibodies. The materials needed for this lab are a reaction plate, a micropipette, micropipette tips, a trash cup for your micropipette tips, simulated knowns for HIV positive and HIV negative, 5 unknown Sera, HIV simulated antigen, secondary simulated antibodies, and substrate color indicator. The procedures for this lab are as follows. Set the micropipette to 25 microliters. Obtain a micropipette tip from the tray. Push the plunger on the micropipette to the first resistance point. Then stick the micropipette into the first solution. Release the plunger to suck up the solution in the micropipette tip. Carefully place the solution in the appropriate well. Refer to your lab procedures for the proper order for adding solutions. To properly place the solution into the well, you must push the plunger all the way down past the first resistance point. Once you're finished placing the solution into the well, remove the micropipette tip from the well and release the plunger completely. Next, and this is very important, discard the micropipette tip after each use. Next, organize your solutions in the order as listed in your procedures. For Part 1, the order for adding solutions is pictured. Refer to your lab procedures for Part 1 to determine which solutions you are to add to each well. For Part 2, the order for adding solutions is pictured. You'll be testing 5 unknown Sera for the presence of HIV antibodies. Refer to your procedures for Part 2 to determine which solutions are to be placed in which well. Finally, determine which of the unknown Seras are HIV positive by comparing them to the controls. Once you are finished, answer your conclusion questions.